Chapter 5 is on homeostasis and transport. Homeostasis, if you remember from earlier, just refers to the stable internal conditions that need to be maintained within an organism. So one of the key features to maintaining this homeostasis is the cell membrane. And the cell membrane gets to control what goes in and out of the cell. It's called selectively permeable. In this first section, Chapter 5, Section 1, we're going to be focusing on passive forms of transport. Passive forms of transport occur without using energy, so no ATP is required in order to have this transport of materials from inside to outside a cell or from outside to inside. There are four main types we're going to discuss: diffusion, osmosis, facilitated diffusion, and diffusion through ion channels. Diffusion is just the movement of molecules from an area of high concentration to low concentration. So you can see in this diagram here, here's our semi-permeable membrane, and these little purple dots are solute particles, or things that have been dissolved in water. The solute particles are obviously much more concentrated on this side of the diagram than they are on the right side. So those particles are going to diffuse from that area of high concentration to the area of low concentration. One term that you'll need to know throughout this chapter is concentration gradient. A gradient occurs whenever there's a difference in the concentration of molecules across a membrane. So if both sides are not equal, that will create a concentration gradient. Now, passive forms of transport are always moving down the gradient. So they move from areas of high concentration to low. They usually give the example of a person standing at the top of the hill with a ball. All you have to do is release the ball and it'll roll down the hill on its own. You don't have to put any energy into that. So it's going from high to low. No energy is required, so it is passive transport. The end point of diffusion is going to be equilibrium. That's where the concentration is equal on both sides of the membrane. So if the particles are diffusing from high concentration to low concentration, eventually they will equal out, and that is equilibrium. Diffusion across the cell membrane depends on the size of molecules, the shape of the molecule, and also its chemical makeup. Some particles that are easy to diffuse are generally small ones that are able to fit through some of those membrane proteins, some of those integral proteins there. And some things that are nonpolar may also be able to easily be dissolved through this nonpolar section of the cell membrane. Okay, that's the lipid portion. So small particles and nonpolar things generally are the easiest particles to diffuse across the membrane. Osmosis is simply diffusion of water particles, so water moving from high concentration to low concentration. In this diagram here, our solute particles are yellow, and you can see that there's more solute over here than here. Here's our membrane. So the solute particles would be diffusing from left to right until they equaled out. But the water is in a higher concentration on the right side than the left. So the water would actually be flowing the opposite direction of the solute particles to try to balance out. There are three types of solutions we're going to discuss: hypotonic, hypertonic, and isotonic solutions. A hypotonic solution means that the solute concentration is higher inside the cell and the water concentration is higher outside the cell. That means that through osmosis, water is going to be diffusing into the cell in order to try to reach equilibrium. So if water is flowing into the cell, that could potentially cause cytolysis, which is where cells could burst. So they're going to fill up with water, and if nothing stops that process, it could eventually cause the cell to rupture. Hypertonic solutions are the exact opposite. Now, that is where you're going to have a higher solute concentration outside the cell, and the water is going to be higher inside the cell. So the water would be moving out of the cell in order to try to achieve that equilibrium. Take, for instance, putting a grape into a very concentrated sugar water solution. Okay. The pure water is going to be in a higher concentration inside the cell than out, or inside the grape than out. So the water will diffuse out of that grape and it will start to shrivel up. And that process of cells shrinking due to that water loss is called plasmolysis. Finally, an isotonic solution is kind of the happy medium. That's where the solute and water concentrations are equal on both sides of the cell membrane. So there's still some movement back and forth, but not drastically like there would be in a hypotonic or hypertonic. So that's really the ideal situation for a cell. Here are just some diagrams of a red blood cell. This is an isotonic solution. This is what the normal red blood cell would look like. There's equal movement of water in and out of the cell. In hypertonic, the water is going to move out of the cell and cause plasmolysis. Hypotonic, water is going to be flowing into the cell and could cause cytolysis. So how cells deal with osmosis? One way is through contractile vacuoles, which are special vacuoles that collect the excess water and then can pump it out of the cell once it's full. Now that process does require energy. That is not passive. Any type of pump that's going to be inside the cell is going to require energy. So that is a contractile vacuole. Turgor pressure is another term that's put in here. Pressure that water exerts against the cell wall is that turgor pressure. So in this diagram here, you can see that the cell membrane is pushing equally against the cell wall in all directions. That means that there's adequate amount of water here in this cell. In this one, you can see plasmolysis has occurred. There's not enough water in the cell, so that membrane has pulled away from the cell wall, and it's not fully pushing up against that cell wall anymore, and that turgor pressure has decreased. That would eventually cause the plant to wilt. Facilitated diffusion is also a form of passive transport, and we're still moving particles down the concentration gradient, so from high concentration to low, but these are going to be usually larger particles that are harder to diffuse normally. So what happens is these carrier proteins that are in the cell membrane are going to help and assist those molecules across. So they can open up. The molecule that needs to be diffused can go into that cell membrane protein, and the protein can seal off to the outside and then reopen on the inside of the cell to release that molecule. Some of those proteins are specific to certain molecules, such as glucose, and then others can move multiple types of molecules across that cell membrane. Finally, the last form of passive transport that we're going to discuss is diffusion through ion channels. And it's very similar to facilitated diffusion, only instead of neutral molecules that we're transporting across the cell membrane, it's going to be ions. So some common ions that we will have transferred across our membrane is the sodium ion, the potassium ion, chlorine ion, and then the calcium ion. And there are more, but these are, again, just the most common. Now, ion channels, some of them are open and others are gated. And what I mean by that is that some of them just have a normal channel that that particular ion can fit in and out of at any point in time. The gated ones are ones that need a signal from the nucleus to tell the cell, hey, we need to open up and bring in more potassium ions. So that protein then will open the gate and allow those ions to flow into the protein and then be transferred into the cell.